You think you've been taught things that aren't quite right? This whole thing about molecules in a puddle or in a pond, lightning strikes, molecules form. Those molecules form into slithering creatures and they come out of this pond. That's the primordial soup model. That's a bunch of nonsense.
So my name is Zach McCrae. I am the chapter director for the Ratio Christi chapter here at Rice. I graduated back in 2020. I was over at Jones. I did a double major, linguistics and theater, and I helped start our chapter here, and I just enjoyed it so much, enjoyed the conversations that we have, that I decided to stay on board. So, welcome. Ratio Christi is a Christian ministry here that likes to tackle the big questions of life. Why are we here? Does God exist? Is the Bible true? And so we enjoy having conversations, having lectures, bringing in subject matter experts like Dr. James Stewart here. Normally, we talk about philosophical, historical, scientific arguments for belief in the Christian faith. But that's not one of those talks tonight. Some of y'all might think that this is about creationism, evolution. No, there is not going to be any God talk here tonight. Rather, we are having Dr. Tour talk about the state of the research on the origin of life, abiogenesis. So, if you aren't a Christian here, we welcome you. And we always welcome you to come to any of our weekly meetings. We normally meet here in this room, 6:30 on Tuesdays. So if you're a Christian, welcome. If you're not a Christian, you are more than welcome to join us. And now let's give a big round of applause for Dr. Tour.
Okay, with intent, there is going to be nothing about God in this. I think I mentioned right at the end, I write the word God to say, you did not hear me speak about God at all. So when people say that Tour uses God of the gaps arguments, or Tour has to bring God into this, or Tour has to bring in miracles into this, they're wrong. Okay? They're wrong. All right, now, I am locked and loaded. And it's not because of you. It's because of my position on these things has brought attack from people on the internet and attack from the scientific community. That's why I am locked and loaded. So if you think that I am being hard on people, trust me, I am an angel compared to the way that they treat me. Okay? So because I know in this day and age, you say one little thing that might, ruffle some feathers, and you look at people like, boy, they should be canceled. But it's going to happen tonight, all right? So there was a video put out a couple of years ago by Professor Dave, who's a guy named Dave Farina, and that has 786,000 views as of today. And he talks a lot about my motivation for doing these things. I was psychoanalyzed, and he knew things about me that I didn't even know about myself at all. And so I came out with a 13-part series. It's actually 14 parts because one of the parts is A and B, but say a 13-part series on abiogenesis. And so you can listen to all of those parts. And so you see, I don't have the YouTube following that he has. This has 43,000 views in the last year. In 2 years, he has 786,000. So, if you guys would just click on it a few times, bring the numbers up, that'd be helpful. And what I also did is, so you don't have to click on all the different parts, you can just click once, and now it's 9 hours of content. And you can just listen to it. Because I had to go through the science. It's just strictly science. I'm going through the science. And there's a lot involved there. And so then he came out with a 2-part series on me again. Now, these have got like almost 800,000 views, 900,000 views. And again, he says I'm still clueless. So we will address that, not so much tonight, but that will be addressed in due time. So it's because I have a day job that I can't just run in and make YouTube videos. And this is not easy because I have to actually read papers and go through the data on the papers. But he brought in, what was really good is he brought in 3 experts And so now I can go after the experts because I'm not going after a YouTuber who it's very hard to engage. It's going like, you put a hook in Jell-O and it comes out. But I'm going after chemists. Only 2 of those people that he brought on knew that they were being brought on. One of them didn't even know. So I was going to go after the 2 today to show really their work on abiogenesis and what it's really like. But I only have time for one because it was so rich. There was so much to go through. So abiogenesis is the origin of life from non-living matter. And to construct any convincing theory of abiogenesis, we must take into account the condition of the earth about 4 billion years ago. These are not my definition. These are textbook dictionary definitions. All right? So this is origin of life. This has nothing to do with evolution, which is life from life. All right, what are the characteristics of life? Responsiveness to the environment, growth and change, ability to reproduce. You have a metabolism and breathe, maintain homeostasis, being made of cells and passing traits on to offspring. Homeostasis is the steady internal physical and chemical condition. All right, again, not my definition. This is what we've got to go after. All right, molecules don't care about life. Organisms care about life. Chemistry, on the contrary, is utterly indifferent to life. Without a biological entity acting on them, molecules have never been shown to move toward life. Never. All right, so a lot of people have tried to make molecules that are reminiscent of the molecules of life. For example, Eschen, Moser, Sutherland, Devaraj, Pauner, Krishnamurthy. And they are origin of life researchers, OOL, origin of life researchers. What they do is they purchase some chemicals, generally in high purity. They mix those together in water in high concentrations, really high concentrations, compared to the way they might be dispersed on an early Earth. Or in a specific order, they attain mixtures of compounds that might resemble a precursor for one of the 4 classes of compounds you need for life. You need 4 classes of compounds: carbohydrates, nucleic acids, amino acids, and lipids. You need those 4 classes. They publish a paper making bold assertions, even though before they do that, all of their compounds are stereo scrambled. They have very little control of relative stereochemistry, no control of absolute stereochemistry, and so they couldn't work anyway. And they'll publish a paper much like Miller and Urey did in 1952. So we're 70 years into this since Miller-Urey experiment, which is a great experiment, I think. It was a great experiment where a bunch of the amino acids formed. So they'll publish these papers, and then the layperson will say, you see, scientists understand this because the press has turned up the knobs, and then they encourage a generation of science textbook writers, and then you hear about these primordial soup models, which are a bunch of nonsense. We have zero evidence for that. The problem with these is this, that molecules decompose. Molecules that compose living systems almost always show homochirality. When you're building molecular systems, you need constant redesigns. So if you hook something on wrongly, you can't go back. Once you hook a moiety on, it's very hard to take it back off again. The synthetic reactions don't know how to stop. Molecules don't know what they're going toward. They don't, let's move toward life today. They don't know that. They're just moving along. And if they make an intermediate that's kind of interesting, kind of in your life, it's going to change into something else the next day under the same conditions. Molecules don't know where to stop. Time, although claimed to be the savior of abiogenesis, is actually the enemy because molecules decompose. You get a molecule, it's good, and then under the same conditions that it formed, it goes bad. All the time. And molecules don't sit around saying, I think I'll sit here for 1 billion years and wait for the next substrate to come. No, they oxidize. And if you say the Earth was reducing, fine. You had a lot of ammonia, which is more degrading than oxygen very often. Prebiotic systems don't have the ability to purify things, and they do relay synthesis. This was the origin of life researchers do. Reagent addition order is essential. It's like making a cake. You can't say, well, let's just put the icing into the flour. No, there's an order in which you have to go. And to build things. You have to control temperature, pressure, solvent, light or no light, pH, what the gas atmosphere is over this. Complex molecule building is hard. The characterization at each step is critical. In biological systems, there's characterization. it doesn't run spectroscopy, but you have enzymes that check things. And if it's not the right thing, then they'll pull it out and they'll chop it up and they'll use it to build something else. Mass transfer is the killer of everything. If you start with a kilo of something, kilo would be a lot if you had a particular thing. And how many steps could you bring that kilo before you would have nothing left? Because each yield might only be 1 or 2% yield. And all of a sudden you have very little material left. And the mass transfer problem, to anybody who does synthesis, you run out of material. You try to optimize material and yields, and it's still hard. Nature keeps no laboratory notebook. So say it's going along, and hey, this is moving like, and now it runs out of material. And it took 400 million years to get there. Well, why doesn't it just go back and make more? Because it never kept a laboratory notebook. You don't keep a laboratory notebook, it doesn't know how to go back and do it again. These are all the big problems that happen. Here's the way synthesis is normally done. You take A, you mix it with B, and you get a number of compounds. A, B will couple together. You get AB1, AB2, you get a number of different compounds. Say AB4 is the one you want. Then you will separate out AB4 from the mixture of compounds. You purify that, and you'll take AB4, mix it with C, then you get AB4C1, AB4C2. Maybe this is the compound that you want. Then you separate this out and bring it on the next step, all right? Then you get the compound you want, you separate it out and bring on the next step. This is the way synthesis is done. Here's the way people who work in the area of origin of life do synthesis. And remember, if you think I'm being hard on them, there's no ad hominem attacks. None. If I hit them with an ad hominem attack, it was not intended. So I'm sorry. I apologize preemptively. All right? But I'm just saying their work is wrong. Their work makes no sense. It's nonsensical. I'm not criticizing the individual. I'm criticizing their work. Direct criticism of their work. Relay synthesis. Here's what they do. They'll take A and B, they make the same series of compounds, and then they never purify this because it's in such a mess they can't purify it. How would early Earth go about purification? Well, it might crystallize. It might. We'll try to crystallize it. Go ahead. Well, they don't. Then they say, we see AB4 in there as a little blip on my HPLC, so it's in there. It might only be 0.1%, but it's in there. So I will go ahead and buy AB4. I will purchase it, and it'll be chiral, homochiral and pure, 100% in enantiomeric excess, or as good as you can get. I will buy it that way, and then I'll go on. Well, wait a minute, that's cheating. They isolated that from a natural source, and then they took it on. And now he or she takes it on, and they get a mixture of different compounds. Say this is the compound that they want. They will say, it's there, I see it. Now they will go ahead and buy this, so they put upon the early Earth constraints that they would never be able to deal with. The heck with early Earth. That's early Earth's problem. Not my problem as origin of life researcher. Not my problem. That's early Earth's problem. You see how unfair that is? It's cheating every step. Nobody could get away with a synthesis like this. Nobody. I mean, reviewers would just laugh at you. But origin of life, you get away with it all the time because they cheat at every step. They cheat at every step. Scientifically, they cheat at every step. Chemically, they cheat at every step. All right, so here's you can just get this off the internet, and this is the prebiotic nucleobases and the routes to them. A lot of this is showing, if you look at these syntheses, they are exceedingly complex, very, very hard, and there is no absolute stereo control. So in other words, not controlling chirality here. In some cases, there's some relative control, but not absolute control. There's no absolute control. There's little or no control of relative stereochemistry. There's little or no accounting of yield. There's little accounting of purification, no accounting for mass transport. They don't say, hey, I ran out of material. Then they look at their notebook and they go back and they make some more, but they don't even really do that because they're only one step back. They just buy some more. It's cheating all the way. A prebiotic Earth would never have these luxuries. So if you just look at carbohydrates, or sometimes called sugars, sometimes called saccharides, and the polymers therefrom. So if we just take glucose, just D-glucose, it can have an open form and 4 closed forms. 2 is 6-membered rings, and 2 is 5-membered rings. There are 4 stereogenic centers, 16 possible isomers on the open form. There are 5 stereogenic centers, so 32 possible isomers. But that's times 4 plus the 16 from here. That's 144 possible isomers just of D-glucose. You already have D-glucose, but it can open or close in, and you can have all of these possible forms. So if you just have D-glucose, how many ways can you hook together 6 D-glucoses? Because you want to make starch, for example. You have to be able to build polysaccharides to have life. How many ways can you hook these together? Well, if you had nucleotides, say you had the A base, there's only one way. A. That's it. 6 A's hooked together. That's the only way. How many ways can you hook together 6 glucoses? It's over 1 trillion ways, over 1 trillion ways to hook this together. And you say, why? Because it's got all these different tentacles. It's got all of these alcohols, all these different, so it can hook together through one or through the other, through an open form, through a closed form, over 1 trillion ways. That's why nobody's ever done it. Nobody's ever made glucose with even relative control of stereochemistry in a prebiotically relevant manner that it is purified from its other material, let alone absolute control where you have to have a homochiral material. Nobody's ever done it. Nobody's ever polymerized glucose. That's how far away we are. Nobody's ever done it in a prebiotically relevant manner. Means that you're not taking human-made design, human protecting groups, deprotecting groups, just what would be available on an early Earth. So, Steve Benner, he's the founder of the Foundation for Applied Molecular Evolution. Why would I talk about Steve's work? Oh, I can't imagine. You look at those videos that came out from Dave Farina, and you will see him saying things about my motivations and why I say these things. And I challenged him. You think that I'm only speaking behind his back. I'm not. I challenged him. We met together. We were at a conference in Israel in May. And I went, as soon as he got to the dinner the night before the conference, I went right at him. And I asked him, I said, did you ever watch my videos? And remember, this is 13 videos. All right, 13 videos of like 9 or 10 hours of videos. Did you ever watch my videos before you commented on them? Because you said that I said many things in there that I never said. And he said, well, it was on, but, you know, I was kind of doing other things. Oh, I believe that. It was on. You turned on part 1. This was long before I launched the click once and watch the whole thing. It was just part. You clicked on part 1, and then you went and did other things, and it was just playing in the background. And then when you got done with that playing in the background, you went to part 2. You clicked on that, and then you went and did other. Yes, I believe you, Steve. Do you believe him? It's hard to believe. Is my voice like soft music in the background that people like to enjoy listening to this stuff? He's published this paper, Prebiotic Chemistry That Could Not Have Happened, meaning that chemistry is so easy, it must have happened on early Earth. Okay, I was interested in this. I wanted to see this. So here's what this scheme that showed in the paper, these different molecules that he could make. And you see all these carbohydrates here, this precursor to a nucleic acid and things like this. Wow, you think that these could happen? Let's look those articles up. So just look up the synthesis of the carbohydrates. So in that article, you have to go back to these other articles to see exactly how did he get this compound? How did he get this compound? So you go and you look up the article. So here's his paper where he talks about the synthesis of this. And so he says, one present obstacle in the RNA first model of the origin of life is the inability to generate reasonable hands-off scenarios for the formation of carbohydrates under conditions where they might have survived for reasonable times once formed. So he understands. He's a real synthetic chemist. I know the person that he worked for his PhD, real synthetic chemist. He knows that these molecules, under the conditions that they're made, they decompose. So how does the chemist make these? You watch the reaction. As soon as it's optimized and starts going up, boom, you stop that reaction and then you fish it out. Because they decompose under the very reaction conditions in which they form. Lots of chemical reactions are like that. But he wants a hands-off scenario. So he picks on other origin of life researchers saying they have too exacting conditions. Early Earth would never have been that exact. Ours is a hands-off scenario. Okay, let's see how hands-off it is. And then he says, here we show how we can do this on a borate material, and it helps the formation, and it makes them stay around longer. It's a hands-off approach. It makes them stay around longer so that they don't decompose so fast. Okay, I want to read about that. Then he shows all I mean, just like, people would be embarrassed to show a mixture like this in a normal synthetic paper. Where are you going with this? But in any case, so he makes his D-ribose. D-ribose has a normal half-life under the conditions in which it forms in 5 hours. That means after 5 hours, you only have half as much as you had 5 hours before. That's what the half-life means. After 8 half-lives, it's gone. Well, he's stabilized this thing so that it lasts 45 hours, which is less than 2 days. When we're talking early Earth and prebiotic stuff, we're talking millions of years. You go from 5 hours to 45 hours, there's no difference. The delta is too small. It's just not there. And this is what you're proud of? That you've stabilized this? What is there something there that as soon as it forms is good? I mean, that's his stability. Now, you look at his D-ribose that he made. This is what the spectrum for D-ribose should look like. This is right off the internet. You guys who are taking my organic class, you're going to learn about C13 NMR by the end of this semester. So you can have an open form and closed form. And so there's D-ribose. Here's what he gets. If you know anything about chemistry, this is an utter joke. That's not a proton NMR. That's a C13 NMR. People would be ashamed to show this. Ashamed. Every one of these little peaks, this is not noise. Every one of these is a different compound. There's a bazillion compounds in here. It's totally useless. And this guy puts it in there as he's proud of it. Somewhere in there is ribose. Now, it's racemic, so you have the other mirror image there. But somewhere there is ribose. Okay? Now what? Now what do you do with it? It's in such a mixture you could never use it. But what does he do? Ta-da, ta-da, he gets ribose. Go on to the next step. Well, where did you get that? Where did you get the ribose? He doesn't say. He says references Krishnamurti's paper. He says Krishnamurti got ribose in his prebiotic work. Oh, okay, so I looked up Krishnamurti's paper. And you know what it says? He bought it. He bought it from Sigma Aldrich. It comes from a natural source. And then he reacts it with this compound. So Benner refers to Krishnamurti. Krishnamurti had taken ribose, which he bought, and he reacted with this compound, which all origin of life researchers say is prebiotically relevant. In other words, it could form on an early Earth. We're sure. And so I searched this thing down. I searched it. So anyway, so where does this reagent come from? So that's also in reference 48. You go to reference 48 and then it refers you to another paper and it refers you to another paper. So you work your way back. And so you can look at here's Krishnamurti's paper. And so he talks about this compound, these compounds that you need for phosphorylating these compounds. And you look up these references. So here it is reference 4 and 5 in the Krishnamurti paper. So he referred me to Krishnamurti, and Krishnamurti refers me to references 4 and 5. So I go to reference, and then he also uses another way. And then if you look at the yields here. His yield of this compound, which Benner is going to use, is 29% yield of a 3-to-2 mixture. Hence, it's 17% yield of what he wants. This is starting with something that he bought chirally pure from a distributor that gets it from a natural source, and he's using all this chemistry that's not really prebiotically relevant. He has magnesium chloride around. So he uses magnesium chloride. I want you to remember that he injects magnesium chloride. He says magnesium chloride, it's going to be available. It is available. 2.5% of the crust of the earth is magnesium. Magnesium chloride is available. All right, so you get this compound. Now, does he take this and somehow get it in pure form? No, it's mixed in with many other things. He has to buy this in pure form or make it by another route in pure form. But nonetheless, where does he get this? This is a hands-off. Remember, Benner said, we do hands-off synthesis. Other people do hands-on. Well, what Benner does is he refers to people who do the hands-on. He says, well, I didn't do it. The other guy did it. Not me, it's him. Here's the hands-off synthesis, D-ribose. Well, where'd you get D-ribose? He bought it from Sigma Aldrich. D-ribose was dissolved in water, and this sodium ammonium phosphate was added. Well, where'd you get that? I bought it. and it was added at room temperature. The pH value of the solution, 9.4, was lowered to maintain it at 7. Oh, how convenient was that? That really happened on an early Earth. Over a 24-hour period by addition of ion exchange resin, amberlite IR 120. Where do you get ion exchange resin on an early Earth? No way. This is not a naturally occurring compound. These are all human-made at 4-hour intervals. Another batch of sodium ammonium phosphate pentahydrate was added at pH 7, maintained as before for 24 hours. Subsequently, the pH of the reaction mixture was lowered to 6.5. How convenient was that? And maintained for another 24 hours by addition of this ion exchange resin. And then the reaction mixture was diluted with 0.05 molar this ammonium formic acid complex. All of this. And then he gets this, and then he has to characterize it. So detailed, so involved. This doesn't happen. Did you ever go out to a puddle and see it doing one of these things? I ask you. This is the hands-on stuff that Benner criticizes, but then he cites this. This is available. It was done in early Earth because here's a paper on prebiotic synthesis where they do this type of thing. And the idea of hands-off is to have someone else do the hands-on part and then cite their product, buy it from an isolator of natural sources, and press ahead. That's what Benner did. Now, when you go to track that compound, which they say is really prebiotically relevant, you go back to this article, which is like from 1955, had nothing to do with prebiotic synthesis, using compounds that would never be available by prebiotic synthesis. You see how the science itself screams out that all of your chemistry is nonsense when it comes to prebiotic synthesis. As for ribose, it was junk, unusable, like everyone else's. No homochirality, so it's not accessible ribose. And even then, it's a trace in a sea of billions of other products. Yet the world believes this nonsense. And I'm the one speaking of miracles. This is miracle right here. This is miracle. All right. So then Benner publishes this paper, which he had talked about on that video. He talked about it on the video, so I go to look up the paper because he's criticizing me and he's talking about how we've made, these polyribonucleic acids. So I go to look up the paper. It's not there. So I contact him. I said, you cited a paper. Could I see the paper? I can't seem to find it. He says, it's not published yet. I said, you talked about an article in a paper as your prime example. It's not even published yet. Could you send me a preprint so I can look at it? No. You'll have to wait till it's published. It came out 13 months after his video. 13 months after his video. And he never said this in his video. It has a substantial fraction of 3,5 dinucleotide linkage. 3,5 is what you want. It has a substantial fraction. What the heck does that mean? Does that mean 2/3 of it? Or does that mean 1/3? Who knows? Who knows? A substantial fraction. He didn't even say the major portion is 3,5, which is the linkage he has to have. So, in a step growth polymerization, and then he goes on in the video about how you can do this with impure materials. That's garbage. You cannot do step growth polymerization with impure materials. There's something called Carruthers equation, which shuts the things down. It says it cannot happen. If you don't have 99.999% pure material, you don't get big polymerizations. It just doesn't happen. So that just can't happen. So here's what it is. If you look at RNA, you want to buy RNA. This is from the chemical manufacturer who isolates this from natural sources. Or they make it on a DNA synthesizing machine. They say you store it at minus 80 degrees centigrade. That's minus 112 Fahrenheit. You have to store it at that with lots of stabilizers to keep it really stable. You'd never want it around divalent cations. So this is the 3,5 linkage. This is what you want. Here's the base. Here's the 3' position going to the 5' position. 3' to 5'. That's how it's polymerized. Here's a 3' to 5', but here's now the 2', 5'. You see, this is 3'. This is 2'. Here's 2'. Uh-oh, that is a wrong attachment. That one doesn't work for coding now into a polypeptide to make an enzyme, to do any synthesis. That's the problem, because RNA can couple through either the 2' hydroxyl or the 3' hydroxyl. And any chemist knows exactly what I'm talking about. And so you look up in his paper, and you click on the experimental of his paper, and in the experimental he put up a Word file, not even a PDF, and it has a different author order than on the original paper. It's really wild. But where did he get his ribose with his nucleotide triphosphate on it? New England Biolabs or Sigma Aldrich, he bought it. Well, why didn't you use the stuff you made? Because it's junk. because he can't use it. He buys it. It's all relay synthesis. It's a game. It's a game. And you're all deceived. All of you. All of you are deceived until 7:30. Then you're going to know. And he says you can do this on impure stuff. Well, then you read his experimental. Before every experiment, glass powders were treated for 20 minutes with 3% hydrogen peroxide. That's really scrubbing it off. Followed by 10 washes with ultra-pure RNase-free water. Well, I could see why you have to have RNase-free because you want to not depolymerize this thing. But 10 washes with ultra-pure water, this could never go on an early Earth where you have normal impurities around. And why is that? Because what happens is that hydroxyl attacks that phosphate and the RNA decomposes. And it decomposes much faster if there's, what is that? Magnesium around. 2.5% of the surface of the Earth is magnesium. You want to be able to add magnesium chloride when you want it. But now when you don't want it, you can exclude it. How did early Earth know that? All right, and then you look in his experimentals and you see that it's got branching, it's got all sorts of crazy structures in here. So the 2,5 could never work. It has been shown by other people that characterize, and they honestly tell you, we made this on clays, and we always get 2,5 linkages. In fact, he says 75 to 80% is the 2,5 linkage. What's wrong with the 2,5 linkage? It doesn't work. It can't code for a protein. So let's just say, let's be gracious and say that he said a substantial fraction is 3,5 linkage. So we'll give him 2/3 of his material. So that's a substantial fraction. 66% of his material is 3,5 linkages. That means the other 1/3 is 2,5 linkages. That means 1 out of every 3 linkages is the 2,5 linkage, the wrong linkage. Well, what does that mean? Well, that means that you could never get even a single amino acid coded for, because each amino acid has to have 3 nucleotides coding for it. If 1 out of 3 has the wrong linkage, you can't even get a single amino acid out of this thing. And you're going around hyping this thing up like it's so amazing. Plus, if you have any of the 2,5 linkage, it acts as siRNA. That's called small interfering RNA. It stops the chemical reaction. Steve Benner starts with pure nucleoside triphosphate. He gets these mixtures. He couldn't even code for 1. So then you see, what do these guys say to the press, which you guys read? And it says, scientists announce a breakthrough in determining life's origin on Earth and maybe Mars. Look, we got to have a cell. We have to have a ton of infrastructure to have a cell work. RNA is not a cell. Even if he had an RNA, and he doesn't even have it. This is so not true. So let's see what Steve Benner says. One community revisits classical questions with complex chemical schemes that require difficult chemistry performed by skilled chemists. That's hands-on stuff. Benner explained. Their beautiful craftwork appears in name-brand journals like Nature and Science. However, precisely because of the complexity of this chemistry, it cannot possibly account for how life originated on Earth. You used it. You used their chemistry in your papers. You see what I mean? He took their hands-on chemistry and applied it, but he didn't touch it. In contrast, the Foundation study takes a simpler approach, and they talk about how they've made these nucleotides 100, 200 nucleotides long in length on this basaltic glass, which is this boron glass. It says the same impactors that formed the glass also transiently reduce the atmosphere with their metal, iron, nickel cores. RNA bases whose sequences store genetic information are formed in such atmospheres. The team had previously showed that nucleotides are formed by simple reaction between ribose, phosphate, and RNA bases. Didn't we just look at that paper? That you cheated all the way along? You never made ribose, you bought the stuff, and you used hands-on chemistry from someone else's reaction that didn't even use prebiotically relevant compounds. This may sound like a bunch of mumbo-jumbo to you, but you're getting enough to catch my drift. The beauty of this model is its simplicity. It can be tested by high schoolers in chemistry class, said Jan Spacek, who was not involved in the study, but who develops instruments to detect alien genetic polymers on Mars. Mix the ingredients, wait for a few days, and detect RNA. No, you won't. No, you won't. If you don't wash that 20 times with ultra-pure water and wash it with hydrogen, you're not getting this. No way. The Carruthers equation tells us that. They say the same rocks resolve other paradoxes in making RNA in a path that moves all the way from simple organic molecules to the first RNA. For example, borate manages the formation of ribose, the R in RNA. It does not. It gave a billion compounds. And that ribose would be totally unusable. You didn't even use it. How do you expect early Earth to extract from that? Benner added, this path starts from simple carbohydrates that could not have formed. He loves that term, in the atmosphere above primitive Earth. Thus, this work completes a path that creates RNA from small organic molecules that were almost certainly present on early Earth. A single geological model moves from 1 to 2 carbon molecules to give RNA molecules long enough to support Darwinian evolution. Is that not enough to make you vomit? I mean, come on. How do you go from small molecules of a bunch of junk that you made, even if you had the RNA that you made? Now you've got Darwinian evolution? So you got life now? And that life is now genetically passing on to offspring? Even Jack Szostak, which himself, we'll see more about Jack Szostak. He's a Nobel Prize winner at Harvard. Here's what he says about Benner's work. If you think I'm being hard, here's what Jack Szostak says to a science magazine. He says, I find it very frustrating that the authors have made an interesting initial finding, but then decided to go with the hype rather than the science. So if you think I'm being judgmental, I mean, he's telling the press this. I'm just telling my community. All right, lipids in the cell membrane. So protocell experiments, they buy homochiral diacetyl lipids and they shake it together. They make these vesicles and they say this is a protocell. It is not a protocell. It is garbage. And the reason is anytime you have a cellular membrane, you have to have different diacetyl lipids on the outside than on the inside. You have to have them different. And why do you have to have them different? Because you have to have a proton gradient to form. Without a proton gradient, there's no life. Plus, you have to have a lot of transmembrane proteins. You have to have glycoproteins on the surface. Cell membrane is really complex. But even just your membrane itself could never work. Nobody knows how to make the outside different than the inside. How do biological systems do it? They have enzymes called flip-base enzymes, and they'll take things and they'll flip them over and move them to the other side. Remember, there's no enzymes there. Chiral-induced spin selectivity. Is homochirality necessary for life? And the answer is yes. Here's what biological systems can do. They can take 2 hydroxyl radicals, and they can have them form hydrogen peroxide or oxygen plus 2 protons. How on earth do biological systems do that? We are just learning in the last 20 years how this is done. And it's because the electron here is spin polarized. It's either spin up or spin down. These can travel down chiral surfaces, and these are spin valves. They only allow one of the electron spins to go through and not the other. And that's how you get the 2 surfaces. One is anti-parallel, one is parallel. It's a spin valve. We don't know how to do synthetic chemistry like that. We don't know how. That's how nature does it so cleanly. So what we're learning is that all of biological systems have this chiral-induced spin selectivity going on, and that's why biological system yields, like in our class, we make 45% of this, 55% of this, 70% of this, and 30% is other junk. How? Why? And in biology, you're getting 99.999% yield or better. And the little bit that isn't right, it has scroungers that go around, find that, and take it out because it would gum up the works. How does it do that? Through chiral-induced spin selectivity. Yes, there are van der Waals interactions, which is this hand in glove we talked about for enzymes, but now you have chiral-induced spin selectivity, which is the thing that dominates. We didn't even know this 20 years ago. I didn't learn this when I was your age. I just learned this 20 years ago. And many chemists don't even know this to this day. And certainly biologists don't know this. The interactomes. If you just look at the protein-protein interactions in a simple yeast cell. Yes, yeast cell is very simple. Just protein-protein interactions. And we're learning that non-covalent interactions have to all be aligned. Non-covalent interactions have to be aligned here. And the reason they have to be aligned because information travels down these by electrostatic potentials. There are 10 to the 79 billion combinations of how these could be ordered. The number of elemental particles is 10 to the 90. You think that's a big number? It's a big, big number. It's a 1 with 79 billion zeros after it. And that's a big, big number. That's the number of combinations. And the yeast cell has the proper combination to get this cell to work. And that's why when a cell divides, it puts this information on both sides of the cell and then it divides. And those do the same thing. The information is constantly passed on. Nobody knows how this is done. This is called the Leventhal 2.0 paradox. Origin of information. Critical for life is the origin of information, DNA or RNA, the order in which these things are attached. This information is primary, matter is secondary. Matter is secondary. So if I have a thought in my mind, all right, and then I write it on a piece of paper. Now it was stored in interconnect pathways in my brain. Now it's written down on a piece of paper. Now I take that paper and I type it into my computer. It's going. It goes into a flash memory. It goes actually into SRAM right away, and then when I hit save, it goes into flash memory. Now I take this and I upload it to the cloud. So it goes through an RF wave to the box on the wall, wherever that is, and it'll go into that box just through an RF wave. So that information has been here. It's been on a piece of paper, it's been on SRAM, it's been on flash memory, now it's in an RF wave. Then when it hits that box, it goes down a wire, that information is going down a wire, then it goes through a server farm into another flash memory. The matter upon which it resides is secondary. The information is primary. The information is the key. And the matter can keep on changing. We don't even know how to make the matter. The carbohydrates, nucleic acids, lipids, and proteins. I can do the same thing with Lee Cronin and his protein garbage that he had in what he talked about. It's all a bunch of nonsense. But this will come out on my YouTube channel, Dr. James Tour. This is coming out. It's going to come out, and all of this will be exposed. All right, a synthetic cell. You say, well, we've read there are synthetic cells. You're not telling the truth. People have made synthetic cells. Oh, yeah? Okay. In 2010, Greg Venter's team copied an existing bacterial genome and transplanted it into another cell. And he called that a synthetic cell. That means that I buy a Corvette. And then what I do is I buy another Corvette. And I take the computer control chip out of the first Corvette and put it into the second Corvette. And I say, bah, I made that Corvette. No, you didn't. You just took something out of an existing bacterial genome and transplanted. 2016, Venter's team did something similar. They removed all but 473 genes from a natural genome and transplanted it into another cell. It didn't work very well. Couldn't reproduce. So in 2021, they added 19 genes back to get it to work a little bit better. And they call these synthetic cells. And people say, Tor, you're wrong. Because I just read a paper where they said, you got to read the paper. You can't just read the title. You got to read the paper. And this is what happens to a lot of people. They say, Tor, you're wrong. You say amino acids don't polymerize in water. I read a paper that says peptides form in water. Yeah, with special activating agents, and you've protected them so you no longer have amino acids. You don't have zwitterionic structures. If you just read the title, you get completely misled because these guys are not to be trusted because they're telling you things that are going to really mix you up. All right, a challenge. Make the simplest living cell. A Nobel Prize would probably be awarded very quickly for the first person who makes a simple cell. Not a complex cell, a simple one, because people will always argue, well, cells were much simpler back then. Well, how simple were they? Well, this has been computed. So bioengineers have computed how simple a cell could be. The simplest cell has to have DNA replication, repair, restriction, and modification machinery, has to have a basic transcription machinery, has to have amino acyl tRNA synthesis, tRNA maturation, modification, ribosomal proteins, ribosome function, maturation, modification, all the way down. And even with all of that, it has to be able to get all 20 amino acids exogenously because it has no ability to make it. It's too simple to make it. This is nanotechnology. This is building systems out of nanocomposite. You have to have all of this in a simple cell. Does anybody have any of this in the junk that they build? No, nowhere close. You have to have all of this. This has been computed. So you can't play this game where cells were really simple back then. All they had was a little bit of RNA. That's a bunch of nonsense. Has to have all of this to be a cell. Remember the requirements that we said a cell has to have. And so go ahead and first you have to make the simplest cell. So first you have to make all the monomeric sugars, which nobody's ever made. Then you have to make all of the amino acids in chirally pure form, which no one's ever made. You can get some racemic, a bunch of racemic amino acids from the Miliuri type experiments and other experiments, but never in chirally pure form. You have to be able to make the nucleotides where you have to first have ribose, then you have to have a base on there, and you have to have a phosphate on there in a relevant manner, and this all has to be chiral. You have to control all 4 of these stereogenic centers, 1 stereogenic center there, 4 stereogenic centers there. Then you have to take glycerol and desymmetrize it, because both of these CH2OH groups are enantiotopic. You say you can't do that? Well, then you can't make a cell. So, nice guy that I am, I'll give you that. Now just polymerize these, put them in the polymers. I'll give you this. Just make the polymers. Just make the polymers. Go ahead, make the polymers out of these things. Nobody knows how to do that. Nobody's ever demonstrated that either. Really, really hard. Remember this? There's over 1 trillion ways you can hook just 6 of them. Now you got to hook like 2,000 of them. All right? How many ways you're going to get that chemistry right? All right. Nice guy. I will give you that as well. And I'll even give you the sequence of nucleotides. So I'm giving you the information, which is another mystery. Nobody knows where this informational code came from. If somebody tells you that the DNA itself is the code, that's a bunch of garbage. That's like saying this memory stick. You know, I just bought it, I have a memory stick in my pocket. So this memory stick, this is the information. I haven't written anything on it. But that's inherently the information. No, the information, this is the medium upon which it's stored. The information is primary. So I'll give you the sequence. I'll even give you that. Even if you had all the components, could you build a cell? Nobel Prize awaits you. Surely you know more than a mindless early Earth. So they can't do it. They can't do it. In other words, even if I gave you a cell, I give you a cell that's alive and it just dies, can you bring it back to life? Because certainly a resurrection has got to be easier than a de novo synthesis. Everything's pretty much in place. Nobody can do it. We don't even know how to define what we just lost when that cell died. How do we even define life? That's how clueless we are. I said the exact same thing I'm saying to you, I said in May with Steve Benner sitting 10 feet in front of me in the audience. You know what he said? Nothing. And nothing is an answer in itself from a scientist. So you say, would you say this to a scientist? I already have. All right. Has the public been misled on origin of life claims? Under simulations of early Earth's atmosphere, scientists have mixed molecules together in the laboratory to produce complex life forms like frogs. 1/3 of the general public thinks that is true. Under simulations of early Earth, scientists have mixed molecules together in the laboratory to produce simple single-cell life forms like bacteria. 2/3 of the public thinks that's true. Who are these people? Well, the average age was 38, half male, half female. 80% of them hold college degrees. You think you've been taught things that aren't quite right? This whole thing about molecules in a puddle or in a pond, lightning strikes, molecules form. Those molecules form into slithering creatures and they come out of this pond. That's the primordial soup model. That's a bunch of nonsense. Here's the dichotomy of the messaging from origin of life researchers themselves. Jack Szostak, Harvard Nobel Prize winner. He said in 2014, he said, we'll have life in the lab in 3 to 5 years. He said that in 2014. How you doing, Jack? How's it going? Harvard astronomer and Origin of Life initiator director, who was also there, Dimar Seselov, also a panelist, told the audience that years ago, Origin of Life was a neglected question, mostly because people thought it was too hard. But outside the meeting room, he confirmed that they were indeed now very close to life in the lab. Seselov thinks it will take like 5 years and not 3. Okay. How's it going, Dimitar? How you doing? 6 months, I don't know if I have it in here, but 6 months after that, Jack Szostak retracted what he said. He said, well, I was being a little overambitious. We're working on making the RNA. You don't even have the RNA yet? Even if you had the RNA, you don't have a cell, you don't have life. He doesn't even have the RNA yet by a prebiotically relevant route. At the University of Chicago, here's what he said in 2021 on a YouTube a virtual lecture at the University of Chicago. And basically, that's because RNA, we know we had to get RNA. And RNA is delicate. It has a phosphodiester backbone. It's subject to degradation by intramolecular attack of the 2' hydroxyl on the phosphate, which I showed you. So you can get cleavage of the backbone, which is catalyzed by metal ions. You have magnesium in there. You probably have a few hours, and it's toast. So we don't have millions of years. Everything has to actually happen pretty quickly. The synthetic processes have to be faster than the degradative ones. He can't even get RNA. And yet he went around saying he'll have life in the lab 3 to 5 years. That's why everybody's confused. Here's Lee Cronin, another origin of life research. He said in this video, September 9th, 2011, he said this: what I'm going to try and do in the next 15 minutes or so, this is during a TED Talk, is to tell you about an idea of how we're going to make matter come alive. And what I'm going to try and do is plant some ideas about how we can transform inorganic dead matter into living matter in inorganic biology. So at the end, Chris Anderson, the moderator, says, just a quick question on timeline. You believe you're going to be successful on this project? When? The question was when. Look at the answer. So many people think that life took millions of years to kick in. We're proposing to do it in just a few hours once we've set up the right chemistry. Chris Anderson says, and when do you think that will happen? Because no answer is an answer in itself. So we pushed him on it. Hopefully in the next 2 years. 2011. How's it going, Lee? This is what I'm talking about. This is the problem. Steve Benner, in 2021, he said to Dave Farina in that YouTube video, I suppose many of the big paradoxes in origin of life have been solved. Like, none of them have been solved. But to the professional in 2019, in a talk, it's all on YouTube, he said, chemistry is actually hard to get to work. The molecules precipitate, the molecules hydrolyze, the molecules decompose. So it's very much a constraint that you have to deal with. It's one goddamn problem after another. I guess I did mention God. Okay? All right? So it's one goddamn problem after another, he says. This is what he says to the expert, because he's a real chemist. He knows we can't even get the molecules made. But that's not what he tells the world. Remember, he's the guy saying, we figured this out. It's life on Earth, life on Mars. I mean, it's all here. Here's 41 textbooks talking about the primordial soup model, the textbooks that you guys grew up with. In fact, these are upper-level college. Here's intro college, upper-level college doing the primordial soup model. You're still learning it to this day. And these are only the ones that I could identify through the help of a friend between 2001 and 2018. This is what you guys learned with. Researchers in origin of life community have publicly accused me of being nefarious and not adhering to current scientific consensus. You bet I don't adhere to current scientific consensus. In fact, some of the biggest discoveries have come when people didn't adhere to the current scientific consensus. So because of that, I'm nefarious, which means wicked, evil. Unlike the claims of making life in 2 years or 3 to 5 years, or that many of the paradoxes have been solved, what really is the current state of origin of life research? Nobody has shown a method to make the homochiral versions of lipids, amino acids, nucleic acids, carbohydrates in a prebiotically relevant manner. Nobody has shown the mixtures found in meteorites or interstellar space could have been useful for synthesis. Even John Sutherland said, I don't think meteorites make the right sort of mixtures, but we need to have more constrained chemistry to actually make the right sort. Little bits of things can be delivered, but they're all mixed in with other things. You can't do chemistry on vast mixtures because they gum up the works. Nobody has shown prebiotic routes to polymerization of the amino acids, nucleotides, or carbohydrates with required specificities that are needed. Carbohydrate polymerization is unfathomably hard. Almost all amino acids need protection. DNA bases sometimes need protection, and you have the 2535 problem. Nobody has solved the code problem for ordering the nucleotides. Nobody has come close to making the higher order structures. Remember, all of these things you have to have a cell. Nobody's ever come close to this. And John Sutherland had the audacity to write in Nature Chemistry, all cellular subsystems could have arisen simultaneously through common chemistry. Boy, I'd love to see that. Nobody has ever shown the lower enantiomeric excess mixtures could facilitate the requisite high-yielding biological-like chemistries with minimization of heat release, thereby mitigating the need for chiral-induced spin selectivity. We do all these computations in our mind, and we dispel 10 watts in the process. This computer is just putting out so much more heat. Why? Why don't we burn up? Why doesn't a cell burn up? Because of chiral-induced spin selectivity. You had to have had chirality from the beginning. It's not something that you could have evolved into. Nobody can explain the requisite interactomes. Remember the 10 to the 79 billion? Nobody, even if given all 4 classes of molecules in any desired order, could prepare even the simplest of cells. Nobody has come close to synthesizing or even suggesting how to synthesize the simplest of cells in a modern laboratory, let alone a sufficient suggestion on how to do this, how the universe could have done it in 100 million years, because that's the time from when the Earth cooled to the time where you find life. As soon as the Earth cooled, you find life. Or even 14 billion years. You want to give it the entire time of the universe? Fine. Nobody has even a hypothetical method for doing this. I've not talked at all about God of the gaps. So when people make that accusation, this is just changing the argument. They want to attack me as an individual because they cannot address the science. Address the science. Address the science. As a scientist, I would never say that we will never understand. I couldn't do that. One day in the distant future, I presume we will understand life's origin. But for now, we are nowhere close. How do I know we're nowhere close? It's because of this. It's because of the moving goalposts. Because every year we find out the cell is more complex than it was the year before. Not because the cell itself got more complex, but we find out about it. We didn't know about chiral-induced spin selectivity. We didn't know about interactomes and the 10 to the 79 billion. And that's just protein-protein interactions. You got protein-DNA interactions. You got protein-RNA interactions. You get DNA-RNA interactions. All of these things going on. Nobody knew about this. So the target is, oh, I got to do that too? So the target's further away. That's how I know we're nowhere close. Okay, therefore, scientists remain clueless on life's origin, and current scientific models for emergence of life are nonsensical. Here's how you can connect with me, and I'll stop right there and open it up for questions. And that's it. Thank you. Yes?
Why do you think there is such a divide between the current state of origin of life science and the public and media perception of it?
Yeah, I think the public has been thoroughly confused, and the origin of the confusion comes with the origin of life researchers themselves. Some argue that the press is responsible. The press is not nearly as responsible as the origin of life researchers. You look at the quotes of these people in their actual papers, it's crazy. And then when they get to the press, it's an order of magnitude more crazy what they say to the press. And it's quotes. So the press is just reporting what they say. And when you do that, you get more money. You get more money for your research because people say, well, you were just on the verge of doing this. That would be a really important advance. So you get more money if people think you're close. If you just say, you know, I just spent a 40-year career doing this, and we're further away than when I began. Who's going to fund that? So, you know, I don't know exactly why they say, but it's from their own mouths, their own words are so off. And that's why the masses are so thoroughly confused. And most scientists are confused on this. Number one, most scientists don't know the difference between origin of life and evolution. They say, Tur, you're always talking about evolution and the problems. I don't talk about evolution. I talk about origin of life. These are two very distinct, different things. Secondly, that they think that this has been solved. I've asked my colleagues, they say, you know, it comes from the foremost reaction. I started looking into this about 6 years ago. Before that, I assumed people had much better models. So scientists, it's not like they're looking at origin of life papers. Nobody cares. They're too busy doing their own things. You know how many emails people get a day? I mean, half your day is just addressing emails. And you don't have time to read origin of life papers. So the community thinks that these origin of life scientists know this stuff. But they don't. Yes?
Yeah, so I guess, how plausible do you think that as we maybe learn more about initial Earth conditions, that some naturalistic account of abiogenesis would be plausible? Or do you think the more we find out, the more challenging we find out that sort of model would even be like probable, even if there's a probability, it'd be
right.
Additional
probabilities. Yeah. Can we find out that the origin of life, the origin of the Earth was different so that the system was better set up to do this? Yeah. It would have to be better set up than the most advanced laboratories in the world today. Because we have no idea. So could it happen? Sure, anything can happen. But it seems implausible based on even the most advanced labs today are clues. We don't know where the origin of information is. We don't know how to keep these molecules stable. So we don't know that. Now, if we can say the life was planted here by some alien, and I'm okay with that. But this is origin of first life. That just begs the question. What is the origin of first life? And that's what we have to get back to. And people will say, well, Tor, what do you think it is? I haven't the foggiest idea. As a scientist, I have no idea. People say, well, then you shouldn't criticize. That's totally wrong. What you do, what we teach our students to do, is you look at a chemical reaction and you formulate a hypothesis on how this reaction may have gone. Then you look at the details. If any of those details say that your hypothesis couldn't be, you get rid of that hypothesis and you think of another. I am shooting this down because maybe one day one of you folks will come with some great advance, but you're not going to come with a great advance if you keep being told we've about got this thing solved. You'll come with a great advance if you say, everybody's clueless on this, and I have a thought on how this might happen. So that's why I'm doing it. Yes?
Yeah, so we have a question online. The first part seems like you already addressed it. Do you consider current pursuits in prebiotic chemistry irrelevant or just overhyped? If you could change in the way origin of life field works scientifically, how would you propose we change the way we approach the question of origin?
Right. Okay, so the question that came in from the internet is that do I think that the work being done is not worth being done, or should we just keep doing it? And what do I propose that we do? I think that the way we're doing it is so wrong. It's just got to stop. Radically stop. It's getting us nowhere. And I say we have something akin to a DARPA challenge. What DARPA does is when they have it, the community is stuck on solving something. So they have DARPA challenge. They don't invite in any of the old guys who've been working on this for 40 years. They want young, fresh minds on this. And you can't do anything that's the same as what's been done, and give us some fresh proposals. That's what we need. Because what I say is all of the origin of life researchers that are working today will die of old age before they solve this problem. And their students will die of old age before they solve this problem. Because I know that we're nowhere close if we keep going down the same way. So I think that there's no use in continuing the same work. We need some totally fresh ideas that are much different than what we're doing today. Is there another question? Yes.
Yeah. I guess I have two questions. One is, could you see like life having some different like structure, like maybe non-carbon based or just like carbon based, but somehow operating differently? And then two.
Wait. So could I see something that's non-carbon based? Sure, you could have silicon. The problem, the great thing about carbon, it forms very, very strong bonds, and the bonds do not easily hydrolyze. When you talk about other elements, water has a tendency to break those up. You talk about polysilanes, you get silicon oxide, which is sand. And you get these very stable compounds that are then dead ends. So the ways you can transform carbon is very broad compared to any other element in the periodic table. And the strength of the bonds, carbon bonds to itself and to many other atoms, very strongly, and under certain conditions, they're modifiable. That's not the case with any other element. That's why there's a whole branch of chemistry called organic chemistry just around the element carbon. So it's hard to envision that, but it doesn't have to be based to carbon. I mean, there's a reason that we eat carbon-based materials because we're made out of carbon. If we were made out of silicon, we'd be eating sand or something like that. We'd be eating something different. Go ahead. Your next question.
Yeah, and then the other thing is, if we look at like processes in the cell, like overall, like how optimized are they compared to like how optimized they could be? Like for example, you mentioned like how our brains are much more power efficient if doing computations in a computer. Is there like cases like where processes in the cell like seem just like so much more efficient that they're almost like ideal?
Are cells so efficient that they can be ideal, these processes? It is very hard to match a cell with many things. But there are human-made things that hands down win. When you're talking about vast computation of crunching of numbers, I mean, computers win hands down over human beings. But as far as the operations within a cell, the making of molecules, it's really exquisite. But it can't willy-nilly just say, hey, I want to put an alcohol there really easily. You don't have an enzyme for that. So you have to genetically modify your DNA to start making some genetic modification that's going to modify an enzyme that'll put a hydroxyl group there, where synthetically I can just go in a lab and boom, just do it. So people are a lot broader in what we can do than a cell is. A cell is very specific to keep it alive. But what it does amazingly clean in ways that we today can't rival. But in 500 years, we may be able to rival. If you asked a man or a woman in the 1700s, when would we have space flight? If they said, I don't know, 300 years, they'd have been right. It was unfathomable in the year 1700 that we'd have space flight. We knew in 1700 that it was not going to happen in 3 to 5 years. We knew it wasn't going to happen. And if anybody said it was going to happen, you'd think back then you'd lock them up. We know we're nowhere close to this thing, but cells are specific. They do a certain job. You can modify them at the genetic level. You can go in and change the genes to change the enzymes to do some modification. But synthetic chemistry that human beings do is a lot broader in what the changes we can make. Yeah, yes, in the back.
So I think one thing that feels about like the primordial soup model is that you don't have to think about it very much, because if you do think about it a lot, then it falls apart. So how would you recommend explaining to people, or actually ourselves, I personally don't have a background at all in chemistry, on a as simplistic way as possible?
Yeah, so how do you explain to people the origin of life in a simplistic way? Because the primordial soup model is so simplistic that it's a convenient thing to explain to people, but it's like totally wrong. It's totally wrong. So, the astronauts were just super good basketball players, and they could jump really high, and that's how they got to the moon. That's a simplistic model for children, but it's so wrong. We would never teach that. We never teach that to children. This is so wrong. It's not worth teaching. I think the better explanation is we do not know how life formed. We just do not know. There are so many problems we do not know. We know the types of compounds that would have to be available. We know that they're going to have a cell and have these properties because life has these properties. We don't know how we arrived at it. And then I think people would be a lot more interested in going into this field. How many people work in origin of life? Not many. Because most people think it's been solved. There was another question. Yes?
Yeah, I was wondering, what makes you personally so passionate and motivated to speak up against it?
Yeah, why am I so motivated to speak up against this? It's because somebody pushed me. Because somebody pushed me. Because what happened was I was going up for an award, and a few of my colleagues said they would never put me up for the award because you have to have other colleagues to nominate you. You can't, I nominate myself for this award. You're not allowed to do that. They said, we won't nominate you because you signed a statement back in 2001, which is now called the Dissent from Darwinism Statement. At the time, I didn't even know what, it came to me in an email. It was like in 2000 or 2001. It came to me in an email. You know how fast you go through emails. And I read this statement. It says, can you agree to this? I said, yes. I found my name on a list with 700 other scientists. And the statement goes something like this, that we find that natural selection and random mutation insufficient for accounting for the diversity of life. Therefore, further research is warranted. Never said Darwinian evolution was wrong. It just says we find it insufficient, so further research is warranted. That statement then was used in the Dover trial, where people were trying to bring creationism into schools, and it was held up as a statement. Look at all these scientists. And so people think that that statement says something that it doesn't. So my colleague said, because you signed that statement. I said, what statement are you talking about? I didn't even know what they were talking about. So then I said, did you ever read that statement? I said, do you know what that statement says? You know what they said? No. Nothing. They didn't know what it said. I said, you go back and read that statement. They came back. They read the statement. One of the people was a Nobel Prize winner. The other was in the National Academy of Science. And they both came back. They said, well, it was carefully crafted. I said, well, duh. I mean, you think they're going to just fly by the seat of their pants on this statement? Where in any other place of science would you not say that further research is warranted? That is the kiss of death for a scientist to say that. We get no more money. Further research is always warranted. But because that came to Touchstone, and they said, because you signed that statement, I said, well, if that's the bottom line, that's the end, I was just minding my own business. Not anymore. And so I started looking into this, and I wanted to track back Darwinian evolution to the beginning, which brought me into Origin of Life. And I started looking into Origin of Life, and I thought, this is smoke and mirrors. This is smoke and mirrors, and nobody knows this except me. Because you have to be an organic chemist to really understand this, number one. Number two, you have to be interested enough to go after it. And number three, you have to be willing to take the shots like I heard came on Instagram when people heard that I was going to speak. Look, they've said all that they're going to say, and that just catalyzes me. It lowers the activation energy to get to the other side of this equilibrium. And you know, every time people say, you know, he's nefarious and he's going against current scientific consensus, I'm like, yes, I'm going to go even more, and I'm going after you. Any other researcher that goes on this Dave Farina's program, I'm going right after them. I'm going to expose their work. Whoever goes on his channel, their work will be exposed. Because they went on a social medium to talk about this stuff, and I will go right after them on the same medium. People say, why don't you just write papers about this? I have. I've written 5 papers about this. People ignore them. People ignore the papers. I am trying to reach the masses. I'm not trying to reach my peers anymore. I'm trying to reach the masses, and that's why I'm doing this. That's why you need to subscribe to Dr. James 2. Yes.
I think what you're talking about brings up maybe a deeper concern about science education and how people are taught that science is done, as opposed to it being this methodology where you ask questions and do experiments. It's, oh, you accept this sort of canon literature, canon set of trust. How do you think we can convince maybe the public that no, like science works as this iterative process where somebody has a hypothesis, somebody points out a problem with this, it becomes further refined, as opposed to just these people are right and these people are wrong because they're in the majority? Well,
this is an area that touches to life itself, so I don't like to psychoanalyze people. I don't like to suggest why they say what they say, because so often I don't even know why I say what I say. You know, and I say things and I walk out of there, I'm like, do I even really believe that? So it's hard for me to psychoanalyze others, but you're absolutely right. The scientific process is different than what young people think who haven't yet been engrossed in this thing. So much is controlled by money, by some powerful people with money, and they can push certain agendas, they can move funding, and if you start bumping up, and you can feel their pain. So when somebody spends, like Benner, the last 40 years of his life working on this thing, and I come along and I say, you don't have ribose, it's garbage. I can see how that would hurt. And so, you know, I can feel their pain. I do. I feel their pain. If somebody were to say, all this time you were talking about graphene, you never made graphene, I'd be like, yikes. What am I going to do now? Either I can just dig my heels, yes, it is graphene, or I can say, yeah, I was wrong. For the last 10 years, I've been wrong. If you like the content that's coming out on this channel, I've not monetized it in the sense of advertising, but if you want to give and you want to help support it, you can give to a 501c3, so it's fully tax deductible, and you can see the link below. We'd love to have your participation, and there's several mechanisms by which you could give. Thank you.